All right, so my talk, standardization of chemical prefixes, CURIEs, URIs, and semantic mappings. This is a talk about what happens when we have all of these resources from different places and they make references to things. And the question is, how do we say a given entity, a given concept, whether it's a chemical mixture, an NMR element of a physical instrument, how do we make sure that we reference those in a standardized way such that we can integrate data across different resources, whether they be ontologies, databases, other kinds of vocabularies, schemas, or whatever. We need to have some kind of standard. So within the semantic web, I'll give a little more preface. Within the semantic web world, we're using CURIEs and URIs for the most case. And so that's what I'm going to talk about in this talk. And I'm going to talk about how the bioregistry is a place that stores the metadata about these things. I'm going to tangentially talk about how the bioregistry is a place that stores all of the other metadata that the last talk was mentioning, and how it pulls in information from other similar registries of metadata that are like partially overlapping yet incomplete, and solves a lot of the issues we just talked about. And then I'll kind of go the next step after we have some tools for making sure that we've standardized the way that we talk about concepts themselves, is talking about semantic mappings, which is when two different resources, two different ontologies, vocabularies, databases talk about the same thing, but they give them different identifiers. How do we deal with this? So yeah, I want to give acknowledgments before I start. First to the URI lab. So I've recently moved with my PI, Ben, who's now a professor. And I want to give a shout out to everyone on our team, the bioregistry team. Big shout out to Philip, who's guided a lot of the things that I've done in the bioregistry and been sort of like a use case driver for many things, and a good liaison for the NFDI project. Okay, part one, prefixes, CURIEs, and URIs. Just to make sure everyone's on the same page about this, I'm just going to give an example of what a URI or like an IRI. I actually don't want to get into the differences between URIs and IRIs today. So I want to show what one of these looks like. This is on the top where we've got this one for the Kebi vocabularies. So all these OBO ontologies, they typically have URIs that look like perl.obolibrary.whatever, and then they've got the name of the ontology. And then after all of that, you've got actually the local unique identifier. So this is the thing actually inside the ontology or the database or the resource. This is not just about ontologies, but this works for any kind of semantic spaces. So you got the local unique identifier comes at the end. Now, typically we want to shorten these URIs because the beginning part doesn't really mean much. As long as we know that this goes with KB, we can sort of shorten that. But the thing is, we need to make sure we know the correspondence between the URI prefix and the CURIE prefix. This is one of the big problems to solve, and it turns out that this is hard for a lot of reasons. The first is that there are tons and tons of different prefixes that show up for the same resource across all sorts of other places, whether it's the first-party resource and how they talk about things, or third-party resources, which might be databases, they might be knowledge graphs, they might be ontologies, how they reference things. So in this example, I'm showing PubChem, which itself isn't an ontology, but it's very important within chemistry to talk about specific chemicals. And depending on what resource you're in or what registry of resources you're in, they talk about PubChem's compounds in a totally different way. Some people, like Cellosaurus, they don't actually realize that PubChem compound is only one of two databases within PubChem, which also includes the PubChem substance database. Sometimes people shorten it, like in BioLink, to just CID. They assume that everybody knows CID means chemical ID from PubChem. So some of them, like identifiers.org, name to thing, prefix commons, they've kind of converged on a standard of saying pubchem.compound using the sort of dot prefix within, or sorry, dot notation within a prefix, this to denote that it's kind of a part of a bigger resource. But then there's other things like ChemInf, Wikidata, and fair sharing that use totally different kinds of references to ontologies or databases themselves. So big problem if you've got queries coming from different places, they may show up with all of these different things as prefixes in them. Same problem for URIs. The part that comes in the beginning could be very different depending on who's using it or why. Even PubChem itself has two different URIs that you can use to get information about a given PubChem identifier. One of them is for looking at their website, and one of them is if you're using their RDF content. This gets even bigger and more confusing because of sort of legacy of identifiers.org is a resource that's supposed to sort of be a redirector on top of all of these other things to make it so you kind of abstract away where the physical resource is on the internet, and then you just use an identifier.org. So they kind of made a huge mistake by making 4 different URIs for everything. And then there's like more and more. I don't want to go into all the possible examples, but the problem is URIs also have a big issue with needing standardizing. So, okay, before we go forward and I talk about the bioregistry, kind of the big questions that I had in mind when I started thinking about this, which was now a couple of years ago, is what are all of the prefixes and the metadata that goes with them? So those are like the CURIE prefixes, the URI prefixes, like the homepage, all the things we just talked about in the last talk. Who keeps track of this information? And so I'm calling it in my talk and the way that I talk about these things, I call a resource that keeps track of information about other resources a registry. Specifically in this context, resources that describe identifier spaces or semantic spaces, which is typically like ontologies and databases and SCOS vocabularies. There was this nice chart that we saw yesterday of sort of the increasing complexity of things. And all of these on a spectrum between, a simple controlled vocabulary and an ontology can sort of be considered something like a semantic space. Okay, and so across the fact that there's lots of different people keeping track of this information, it maybe is partially overlapping, but there's lots of missing bits to it. How consistent are the prefixes used in each of these? How good do we cover our domain? So today we're just talking about chemistry, but the thing that I'm going to mention, the bioregistry, it's sort of generic towards life sciences. And in fact, the code and the infrastructure that underlies it can be redeployed for any domain, which also includes something like engineering and may be relevant for other NFDI consortium members. And can we do better? I think I should take this off my slides because I was sort of using this at the beginning when I was trying to push the bioregistry as sort of a new resource. And I think it's now, we have done better. So let me sort of get to it and show you what is the bioregistry. All right. Before we get to that, I have one more slide. So I mentioned that there's lots of different registries that kind of have different scopes, different domains. I mentioned identifiers.org, named a thing and prefix commons. These are all resources that are trying to be resolvers that keep track of the URI prefixes. So you can take a given CURIE, like pubchem, pubchem.compound : 11000101, and then turn that into a web page you can look at. But there's also other things that we've talked about, like ontology registries, like the Obo Foundry, Bioportal, AgraPortal, all of the OntoPortal instances, also AberOwl, and some other lookup services that kind of allow you to index those. So actually the TIB service lives in this sort of lookup service or ontology registry space. And then there's like database specific registries where resources like Uniprot and the Gene Ontology, they said we're going to maintain our own version of this because we don't want to have to align on anybody else. Or actually, more likely, they started doing it before there were any shared resources that they could use. And then there's kind of some other random stuff like fair sharing and Wikidata and BioLink, which are all very specific and not generally useful. So we can pull parts of them to accomplish what we want to with the bioregistry. So I mentioned in the comments that a comparison of sort of the capabilities, the metadata schema of each of these things that I've mentioned, how they all kind of stack up. There's actually a thing on the bioregistry website for this. So I put it in the comments in the last talk, but it's also here if you want to take a look at how these things all compare to each other, and also the bioregistry itself, which I'll get to momentarily. So yeah, the issues with previous registries, the different scope, the metadata standards, topic areas, the fact that they're incomplete. Some of them are sort of, not curated anymore. Identifiers.org is a pretty good example of something that doesn't have a lot of curation effort anymore. So this has a problem with maintenance. Project governance is an issue with a lot of these things. Like if it's curated internally by a small team and it's done in a proprietary database or like an internal database, then it makes it not possible for external people to contribute and to help maintain it. Content not aligning with other registries is like a minor issue because that can be a technical solution. And then also the other thing to mention is like some of these resources are not licensed in a permissive way. So I poke fun at fair sharing itself for being a resource that's prided on fair, but it's actually not that open because it has this SA share-like clause in its license. So that's an issue for reuse and redistribution and derivation. Okay. So what is the bioregistry? The solution to all these things, it's a registry of all the prefixes and the associated metadata we heard about in the last talk. I think for the most part, I cover almost all of these things within the bioregistry. And then it also provides this de facto standard for what's the preferred or standardized CURIE prefix and URI prefix you should use when you standardize your data. The bioregistry sort of goes further in its concept than the other registries by having this meta-registry concept, which actually maps the prefix in this registry is the same as the prefix for this in the bioregistry, and it becomes the hub. So if we go back a couple slides, this is the hub and spoke model where the bioregistry is actually mapping to all of these other prefixes. Okay, so the bioregistry also has this resolution capability. Once you have all the information about URI prefixes, then this is a relatively simple thing. And the important thing about the bioregistry, on top of all its capabilities, is sort of its governance and its forward-looking nature of being completely open code and open data, using permissively licensed code that anybody can sort of make a pull request onto or look at or modify, and then to use infrastructure that's open. So we use GitHub Actions and GitHub as a continuous integration and continuous deployment service to make sure that we can quickly make updates, and that this sort of abstracts away this issue that Philip mentioned with, like, what happens when funding ends, what happens when key people move on. Like, this resource should be resilient to that, because anybody can sort of pick it up and continue curating it. Yeah, and so the way that works sort of from a social level is that it has governance models that have contribution guidelines to train people on how to make contributions, but also a harness to help people make contributions in a quick and easy way. I'm going to show you how to make this contribution later, where we're going to put the voc for cat vocabulary into the bioregistry together during this talk. And then having project governance that says who gets access to this repository. If the people who are maintaining it now, we say, okay, we're moving on, we're working on different stuff. Somebody else should be able to make pushes from a technical perspective. So this is all stuff I've talked about in this other talk that Philip was mentioning. So sorry for everyone that's kind of like a little bit jarring. I can make a link to that talk about how do we make resources more sustainable and resilient to this sort of thing. If you want to look at the bioregistry, there's links down here for the website, the source code. It has a Python package that goes with it that helps sort of operationalize all of these things. And then let's kind of talk about, I'm going to skip this slide. I'm going to give you not a live demo of the website itself, but I'll show you what's inside the bioregistry. So the first thing is there's a page that has the list of all the prefixes, and you can search it, and you can find new chemistry terminologies you didn't even know about. You can find all sorts of things that aren't ontologies. So this is the place to start. On each ontology page, you have all this metadata. So this goes with a lot of the things that we were talking about before. We've got version information, licensing. We've got links to the homepage, the issue tracker, Git repository, the OWL download, who the contact person is, how identifiers in this resource look, publications, ways to regular expressions so we can validate them, this kind of information. And so we've got a web page here, but it's actually also the case that you can download all of this from the API. You can also use content negotiation to get this in various linked data formats, and it also comes with bioschemas and schemas.org schemas built into it already. So this is indexable. So I mentioned before, the meta registry is also part of a page for a given resource or a prefix. So this cross links to all of the other resources. So I'm using cheminf as the example before, and you can see that this is a bunch of other registries that also index cheminf, and this is the way that they index them with these various other prefixes or keys. And then there's also to some extent information about how one prefix is related to other prefixes or the resource that the prefix is attached to. So for example, cheminf appears in the Proco ontology, and it's also in this collection, NFDI for chem. So what are collections? I think this is a great place for us to group together lists of related prefixes. So Philip and I have put together this NFDI for chem collection that sort of covers all of the things on this page, the ontology collection page from the NFDI for cat website. And so this links to all of the prefix pages for this. And this is a nice place that we can put all these things together. Okay, two other things built on top of the bioregistry. First is the ontology quality assessment website. And so this is a script that runs on a periodic basis, and it downloads all the ontologies that are listed in the bioregistry. And then it goes through and it checks all the references, and it gives feedback hey, these references have prefixes we've never heard of because they're using CURIEs, or these URIs we don't know how to parse, and this is a great way to arbitrarily make some assessments on the standardization of a given ontology. So here's an example of eNanomapper, and so we've got some references that don't really look like they make a lot of sense. So this would be a tool that can be used to make some assessments of the standardization without actually having to use any of the bioregistry code yourself. This is built on top of the various parts of the bioregistry database and the code built on it. And so as we add more NFDI ontologies, this ontology quality assessment automatically builds this website using GitHub CI, and this is a way that we can point to give feedback to curators and say, hey, maybe you want to address some of these things to improve sort of the content of your ontology. The next one is also built on bioregistry. This is the OLS standardization analysis. So there's various OLS instances floating around in the web, and this is just another tool that goes through and it checks all those instances. And the the resources inside them, to what extent they are mappable to bioregistry. So the TIB instance, we've got 31% of the prefixes in the TIB instance are mappable to the bioregistry. This is for a couple of reasons, like only a subset of that is relevant for the chemistry use case. And the bioregistry has, this kind of life sciences domain scope. So there's all sorts of stuff inside the TIB instance that actually isn't relevant. So we can make a little bit of improvement on the way that it's calculating this number, but this is also a good dashboard to ask the question, hey, have we got all of our resources within the NFTI project standardized against the bioregistry? Okay, so that's the end of what I have to say about the bioregistry. And I have like kind of a 6-point call to action for NFTI for Chem. What would I like to see all of you doing with this if you have the time and the interest? First, I'd love to see all of the ontologies you're working with, register them in the bioregistry, and not just ontologies, other controlled vocabularies, other semantic web artifacts, that sort of thing. We're going to do this together in a live demo in a minute. Next, for all the ones that are already in the bioregistry, which by the way is many of them, you saw in this collections list before, almost all of the ones from the NFTI for Cat list are there. So when you're going through, if you find that there's more metadata you know about that isn't in the bioregistry, this actually means that this metadata isn't just not in the bioregistry, but it's not in any of the other resources the bioregistry integrates. So please contribute. Use the bioregistry to find new resources that are relevant for your project. And help me make, actionable collections that correspond to NFTI for Chem, NFTI for Cat. Even to some extent, the bioregistry may be deployable for other domains. And so if that's interesting for the engineering group, please let me know, and we can work on that together. This is an optional thing, but the OLS configuration is based roughly on the Obo Foundry configuration, which is kind of a subset of what's in the bioregistry. So you could potentially use these collections to seed your OLS instances, sort of reduce the kind of effort and configuration it takes to put those together. So that's kind of like a pie in the sky idea. And then like I mentioned before with the ontology quality assessment, use the bioregistry to validate the content of your ontologies and to help make a conversation with the maintainers and the developers of these ontologies and to make them better. Okay. Do people have interest to see a live demo of like adding something to the bioregistry? Otherwise, we can sort of jump forward to semantic mappings. And also, let me double check how much time I've got. I guess I have 10 minutes.
Yeah, roughly, but rather more 7. So if we want to have some questions later on, I would say, unfortunately, luckily you gave quite a few talks. So let's skip the live demo and rather go to the semantic mappings because this is also very important.
Okay, good. Yeah. So I have some things on YouTube that show how to do this sort of registration, and there's some talks on this. So let's move on. So semantic mappings. What happens once you already have a really good and consistent way of referencing things? And I'm going to go through this in absolute lightning round because I do realize that we started a little bit late and I want to be respectful and stay on time. So I'm going to try and do this in just a couple minutes. So semantic mappings, we've got something in KB, we've got something in drug bank. They're both identified by their own CURIEs, but they're the same thing. So they're equivalent. There's all sorts of different kinds of semantics between how semantic mappings are related. Sometimes they're broader, sometimes they're narrow. There's SCOS vocabulary for this. I would definitely point everybody to say, look at the SESM 2023 slides for more details about like what mappings are and a more principled way of talking about them. I'm going to skip this slide. And use SESM. So I also say you should check out Nico's slide deck on using SESM as a place to store mappings in sort of a high quality way. This is a standard I think the ontology community is starting to align on. So use this. So the problem is we've got like, for me personally, I want to map data and knowledge from different places. And like this is an example with cell lines, but it turns out that there's like all sorts of cell line vocabularies. And there's only mappings available from first party between like certain vocabularies. So there's CCLE and it maps to DETMAP, and DETMAP maps to Cellosaurus, and Cellosaurus maps to DETMAP and to Brenda and EFO. And then there's cell ontology, which only maps to Brenda. And I have data that's in CCLE identifiers, and I have knowledge from my text mining system Indra, which has got cell ontology identifiers. I'm like, how do I map between these two things? Actually, I want to take advantage of all the mappings that go across all of these other resources and do inference, but like even then it's still incomplete. So there's a big mess of different resources, and we want to make sure that there's full mappings between all of the entities in one resource to all the entities in the other when possible. So here's how that looks. Like if you do it, it turns out that there's actually incomplete mappings between all of these, even if you use inference. So we want to be able to deal with some of these issues. And then the last part is automated assembly. Let's say I download Kiro, which has like really interesting relationships between chemicals that have CURIE identifiers, and then also download drug bank, which has interesting relationships, but they have drug bank identifiers. I really want to be able to like combine my nodes together, and I want to like use CURIE, for example, here. And so then I have all of the relationships are sort of remapped. So I only have one node in my automated assembled knowledge graph that has a reference to ethanol. Okay, so problems. First-party resources usually have incomplete mappings. People make predictions all the time, but they usually don't persist them and usually don't curate them. We need a curation tool for predicted mappings, and we need to like reincorporate and contribute curations, upstream. So we want to be able to make sure that once we do this stuff, that it gets back into the ontology so everybody can use it. And I pick fun at the Ontology Alignment Evaluation Initiative, which for more than 15 years has been doing the same ontology alignment task. I say, why didn't they the first year curate all the results and then sort of move on? Yeah. I don't know why they didn't do this. So all this needs to be done open code, open source, open data. This is 03. Maybe you'll hear about 03 in the future. So we want to make sure that this is sort of sustainable. The biomappings, it's bio everything. Biomappings curation cycle sort of is a workflow and a technical backend to doing this, where you can sort of first retrieve resources, make predictions, filter them based on whatever your domain is, do curation of semantic mappings, and then improve the whole process with whatever wisdom you gain and whatever negative mappings you curate that are incorrect. I'm going to skip talking about the methods itself, but one of the best ways to do mappings between different resources is like lexical alignments. So yeah, we can find a lot of the same resources appear in different places, actually with the same name. So here's a 3,4-dihydroxy benzoate showing up in CURIE and mesh. And so these are equivalent. We've got small label differences with capitalization that are different. We've got sometimes when we have synonyms in one resource, they match the primary labels of others. And then we've got sort of the biomappings curation interface, which is a place where we can do this. So I was going to give a live demo on this too, but I actually don't have enough time for that. So this last slide is something you can follow if you want to do some biomappings yourself. And the idea is you get this curation interface and you go through and you give a thumbs up and a thumbs down as you're curating for things. And you can search for the namespaces you want to work with. So if you want to do CURIE mappings today to mesh, you can search for CURIE. And then at the end, it sort of integrates with your Git system, and then it allows you to make pull requests. And then the results from this, they go to the internet and everybody gets to use them. Okay, so this is kind of what it looks like in the end. The call to action for NFEI for Chem is generate predicted lexical mappings between your interesting ontologies. So like yesterday, we talked about how there was this measurement datum that was appearing in, I guess, in OB, and then there was also something in CHMO, and they're the same thing. We can sort of generate mappings between those things, and then we can help resolve some of these informations. So this is a short version of talking about biomappings, but you can use that to do that curation. And then I'm also kind of thinking about different ways to automatically assess the landscape of many different resources that all live in the same domain and how much overlap they have, sort of like a more automated version of this very fast image I showed you with all the cell line mappings. So if you want to work on that with me, let me know as well. All right, that's it. Thank you. I'll take questions. We're just at the top of the hour, so I think I managed to do this in like 25 minutes or so.